Ladies and gentlemen, welcome to the second session of the celebration of the 150th birthday of the Zoological Station. I have an announcement to do because Tim Hunt, the Nobel Prize Tim Hunt, who was in our scientific council for 2 mandates, no, maybe 3, has COVID, and he doesn't have COVID like Christiane Gröben, who didn't come here because she didn't want to infect us all, but you have seen yesterday she's in mint state. Tim is sick in bed, and so he cannot even deliver his talk from remote as it was supposed to do. He would deliver it from his bed, and I don't think that this is fine. So we give our best wishes to Tim if he's watching us from remote. I don't think so, but anyway. So we start with the second, that now becomes the first presentation. By Wiebe Koistra. I have to say that Wiebe was complaining before because the font used for the program is microscopic. So you need a microscope to read the titles of the presentations. And so I will not read the title of the presentation because it will be there. So Wiebe, the floor is yours. You have 20 minutes.
Thank you. Now I see how this works.
And then you can, bravo.
Thank you. Well, I thank you for inviting me here, and I know that many people are watching this in remote, so we are in a sort of hybrid condition between the normal world and the COVID world. But COVID is not over yet. Anyway, my talk. Yeah. No. I have to push very hard. No, it is fine. It's fine. I just have to use violence. Anyway, so I am here, Wiebe Koistra, since 20 years or more at the Stazione Zoologica, but originally Dutch. Now in Naples and adopted by the Napolitans, and according to some of you, become more Napolitan than the Napolitans. I don't know if I have to take that as a compliment. Anyway, I would ask you to just imagine you are in a forest. And so the brown, leathery foliage is overhead, and green sunlight shines through this foliage onto the forest floor, which is a riot of red and pink and orange and yellow and all the colors of the rainbow you can imagine. Now, if you think that comes out of a Beatle song, no, this is marine biology. There we are. The first time I swam through such a forest, I was totally fascinated. I thought, this is beautiful. I was enthralled by the richness of the algal flora there on the bottom, and it was there and then that I decided that I wanted to become a marine biologist and that I wanted to study algae, become a phycologist. The second important point for this talk is this happened at a marine biological station. Not the Stazione Zoologica, but the competition in Roscoff, the SBR. But it doesn't matter for the whole case. In any case, I never looked back, and my career was in phycology. So I think that many among you in the audience have gone through a comparable experience. It may not have been with phycology, but it may have been with something else, and that career choice defining moment was there and then, and you probably remember. So I did. And so these people did. Now here you see a group photo of a bunch of early career scientists. they're still in their young years, so some of them went on to become very, very, very famous, but there they are, just the wild young men and women they were when the photo was taken. And there is, however, an important difference. And all of you spot from your own perspective that important difference, but I tell you what I think, for the matter of this talk, what that important difference is. Namely, the people on the right had a fully equipped marine biological station to their disposal, and with all the equipment and all the service personnel that they needed to get their work done. So they are there happily ready for a photo and then work out the results with microscopes and everything. Whereas the people to the left, a very similar group, very similar spirit, had to carry all their research equipment by themselves. There was no marine station at that time. And some of these people had a thought, they had a vision, hey, wouldn't it be nice to have marine stations? And the result of that you can see all around you, and you see here the Stazione Zoologica and some other famous marine research institute. Also our guest from Plymouth will recognize his own building. And so there are several others, and you may have seen them anyway. So the spirit of that time was, of course, not only here because Roscoff was founded at about the same time, but it was with the same spirit and with the same choice to study marine biology. Now, that's one phase, so we have now all these buildings all over Europe and all over the world. Now one step further. Because Dohrn had a vision, he had some more vision. What if a scientist could come to one marine station, make use of the research infrastructure there and all the personnel and the help and the scientific staff, and then take the data and work them out, and then hop on the train off to the next marine station, do some more work there with the local help of the staff, and then work out the data in collaboration with the local staff who is expert on all the stuff. Wouldn't that be great? And of course, the pictures here are a bit off for Dohrn. If Dohrn would have seen this, he would have been flabbergasted, shocked, but the spirit is the same here again. Now, what I want to illustrate here is, in order to have that kind of facility of traveling around at ease, you don't need just a network of marine stations. You need something more. You need the administrations to work together. You need the funding streams to work together. You need the stations to collaborate, to cooperate in a more profound way. You need to make choices. We will do this here if you do that type of work there, so that people have also a reason to travel. And so that kind of vision is not so easy to realize. So Dohrn may have had that vision in the 1870s, but I think that Queen Victoria here expresses it all with her facial sort of Whereas nowadays, hey, this is Europe. Now I have here a little footnote. I prepared this slide in January. And that was the time that we all thought, oh yes, we don't have war in Europe. We are now collaborating, we are now collaborating all, and the borders don't exist anymore. And then came the Ukraine war that showed us that things are not that kind of happy rosiness. But at least within the blue bits, we now all collaborate together, and we have this supranational organization that allows us to work across Europe. So the first thing that Europe stimulated was this kind of networks of excellence, where marine stations and research institute with scientists in marine biology collaborated so that the researchers could start networking, develop all sorts of things together at a European scale. And lots of stuff came out of that. But the problem with these networks is that they have a restricted lifetime. So after the funding from Europe finishes, and plop, everything sort of collapses, and it becomes a struggle to maintain the things that you developed. The next step for Europe was to organize the research infrastructure, so in this case the marine stations, to come together and organize a transnational access program in the spirit of Anton Dohrn, to have people from all over Europe come to marine stations to do work, all expenses paid. And that is a wonderful. We have had two phases: Assemble Marine, and then there was a second installment, and that is Assemble Marine Expanded, and that expansion was with marine stations of EMBRC and other marine stations all over Europe. Also here, this is, I think, a reasonable picture of the Mars network. So that also has a restricted lifetime, because when this project finishes, European projects last some 4 or 5 years and then it's over. So the next step is that Europe came to the conclusion that we have to have something more permanent. Now, and that is the European Strategy Forum for Research Infrastructures to plan and organize this. And what did they do? They were aiding the establishment of European Research Infrastructure Consortia. And what are those? In short, ERICs. Those are multinational consortia of member states. I stress this because they are the paymasters, so they have to guarantee that things are paid for a very long time to be, and not just for 5 years. They are operating advanced research infrastructure. Now, what's that? Well, very large marine biological stations with all the toolkit is there. That's a typical research infrastructure. It enables researchers to have open access. It doesn't mean that the researchers don't need to pay anything. Of course they need to pay. They need to get the funding for it. But then, when they have the funding, they can come to the marine stations to make use of whatever they need. So it is not a free loading. The funds for the preparatory phase come from Europe because they want to gesteren, they want to make this happen, but then the member states have to commit to long-term funding of those ERICs. It's a little bit like EMBL or the European Space Agency or the Large Hadron Collider. So these are examples of research infrastructure that can be centered in one place or distributed all over Europe. And marine stations is an example of all over Europe. So here are a flock of all those marine and non-so-marine but biological research infrastructures that sprang up, various different initiatives. And one of them there in the middle is EMBRC, the European Marine Biological Resource Center. And this is what we are now. I'm skipping here over all the wonderful history because I want to go into the future. Anyway, there's nothing wrong with history because these things were built by many different people that have now gone retired or have gone all together. But we are now here with 10 countries in Europe and also Israel. And EMBRC headquarters are now at the Sorbonne University in Paris. And it is the member states that pay for the long-term sustenance of this system. And you see also Italy is in, and the Stazione Zoologica is the headquarters in Italy, so the node, and several partners in Italy are in this as well, such as the CNR, the various different universities with marine biological departments, the OGS, and a couple of other research institutes. So we have a very diversified offer of services. Now, what are the overarching aims? There are, of course, lots of aims, but I focus here on the essentials. To provide researchers and industry with access to marine biodiversity and ecosystems. And, most important, also the tools and services needed to do that kind of research. And that's what we are here. And so there is one other factor, it has to be sustainable. We are not selling out marine biodiversity to industry. We do that if we want to give industry the things to develop further. It has to be done in a sustainable way. So we develop something small and then they can make it into something big. It is not that we are harvesting the place here clean because some industry needs some material. I stress this, no fear. So then there is this diversified system, and I think I talk here to most of you as marine biologists, but some people don't really know what's inside a marine biological station, a large marine biological stations, because there's several functionalities in the services, and one of them is ecosystem access. Now here you see an example of such an environment, and the toolbox that you need to study that environment. And you see there are all sorts of facilities listed, and it is the personnel of those facilities and the maintenance of those facilities and the fact that those facilities can be used safely that makes a marine station to what it is. So imagine you use an ROV and you don't know how to use it. Within 5 minutes that thing will be wrecked on the bottom. So you need the expertise to make use of it. Then there are the biological resources. Now that's culture collections, collections of preserved samples and so on and so forth. There is an example. But these biological resources are not just there. What they need behind that is the taxonomic expertise to key everything out and know what is what, so that people can make use and know that the species they use are authenticated. And of course there is the personnel that needs to maintain these facilities so that they are available for use. Experimental facilities. Now what's that? Of course that is a whole lot of aquaria, endomesocosms, laboratories, climate controlled rooms and so on and so forth. Also there are personnel who knows how it works and can help you making use of it. And crucially, when you do want to set up a research project and the instrument with which you want to do that doesn't exist, it has to be designed. So there should be technical personnel that is very skilled in designing machinery and toolboxes and toolkit that doesn't exist and that they have to design with you. They have to discuss. The first prototype doesn't work, the next prototype will work, and then you can do research that is on the cutting edge of science because nobody has that kind of equipment. You made it new and there is possibilities for patenting it. And there is a box of things that is called the technology platforms in the parlance of EMBRC. Those are instruments like the imaging, the microscopy, SEM, These are not typical for marine stations, but you need them anyway to do research, so they have to be there as well. And then I go on, research develops lots and lots, megatons of data, and all these data need to be managed. And that is a relatively new thing, that is the e-infrastructure data and services that have to be present. And you see here, so people have to have access to data, they have to know how to do that, so you need a high-performance computing to analyze this stuff, and then the data analysis and the data management and the verification so that you can reuse data and that the data are findable, accessible, interoperable, and reusable. That's what FAIR means. So all that and a lot more, outreach, training, and so on, are all part of marine stations. I cannot be, in 20 minutes, I cannot tell every single detail, but you know that it is there. But the question now comes, why would you share this with external users coming from all over the place? Well, there is a very good reason for that. Because advanced research infrastructure, as we have it here, is very, very expensive to maintain, to buy. It is unaffordable if you don't have the money for it. But it's often also underused, and that means that the fixed costs are divided over just a few users, and that makes it outrageously expensive for those who want to use it. We would not be able to afford a submersible, But if we share it with other people, the fixed cost per unit of use come down, and suddenly it becomes affordable. Of course, we have to write in Lab Agenda when we want to use it. We cannot say, tomorrow I want to use it, let it be available. Why is it not available? Because somebody else uses it. So you have to arrange yourself. But there is another thing. Access to external users fosters, of course, exchange, collaboration, and things published together. And that also counts. Now, why share this across borders? Of course, there are different marine ecosystems across Europe, and if you want to study laminaria fields, you have to go to Messina, or you have to go to Western Europe. You cannot do that here. But that's not the main point. The main point is that each marine station has specialties. It cannot afford every single thing that is needed. So in some cases, you just better go to another place, and your project or the marine station where you work pays for your access to that other place, because then they don't need to afford that expensive instrumentation and personnel here. You can just go to another place. And other people from elsewhere come here because we have an excellent, let us say, submersible, which you can study the Canyon Dohrn, or we have this ecosystem in Ischia where there's the bubble site where you can explore the future ocean. So there are all sorts of reasons to collaborate in this kind of things, and not to mention that some kinds of things are better centralized in one place with all the expert staff there, because we can connect with it via the internet. Now, I want to come to the finish of my talk to explain to you that 8 times 1, if collaborating, is far more than 8. What's this here? This is a Chinese legend of at least 2,500 years old, if not more, where 8 immortals, 8 superheroes in modern parlance of Hollywood, come all together to put their skills and their magical toolkit together to do something that is considered absolutely impossible, and that is traversing and exploring the ocean as they go. And that's what these people are doing here. Now, of course, the modern times, there are no magical superheroes among us. The team of people who work on making EMBSE possible are just human beings, no superhuman skills. But what they do is they happily collaborate. Here, this is the so-called committee of nodes, with representatives of each member country, and we work happily together. And we speak all the same language, different languages at that, but that is fine because we can communicate. And we can do things together and make this all work to build this research infrastructure. And sometimes I have the feeling that, of course, people come and go in this team, but the spirit carries on, and different people in different times put their shoulders underneath it. But I sometimes have this feeling that we are being cheered on by these people from the past who joyously and happily say, yes, okay, carry on, this is always what we have dreamed of. And please continue this good work, because this is an excellent idea to carry forward in the future. We always have thought that. And thanks.
Thanks, Wiebe. And now I give the floor to Roberto Bassi, who happens to be the chair of our scientific council, and he will tell us about the potentials of biotechnologies.
Thanks, Nando, for the presentation, and I really am happy to, after many years being serving in the scientific council of the station, to tell you something about my research. And in fact, I have chosen a different angle with respect to the general tone of the presentation. I will do the other way around as respect to most of the other presentation, which consists in going not for the basic research to apply the research, as is the usual way, but the other way around. So to see what the applied research can bring to fundamental studies of marine and aquatic research. So, in fact, my lab in Verona has been developing mostly basic research. But during this time, we had to cope with applied research. And that was surprising how much that helped us in setting technologies for being back to problems that we have been unable to solve before, and in fact being able to approach those projects. So the marine biotechnology is very important, and the first part of my talk will be telling why this is important. So in the general balance of energy in this planet, photosynthesis is extremely important, and algal photosynthesis and plant photosynthesis brings all the energy in this planet, producing fresh biomass and food, of course. But this production of food has been limiting so far for the development of the human being. And so far we have been able to cope. If we take 100% the production in 1960, we are now, we have been increasing by 60%, so we are 160 now. But by 2050, in which we will be around 11 billions, we will be able, with the present technology, to obtain a rise to double the 1960 productivity and go to 200. But what we would need with the trends that the actual population is 360. So now we face a problem with the energy, but the next problem, very close, will be a shortage of food. Of course, not only food is important, but also energy and fuels that we already know that is a major problem. And now we see that the fossil fuels also come from photosynthesis, but some time ago. And there is the possibility to produce fossil, to produce the fuels from fresh biomass. If we can produce so much biomass to spare beside the food for that. This is a possibility that will contribute a lot to sustainability. So we have to take in account the need for human being, and we need to preserve the environment. So this is because the production of biofuels is carbon neutral and is a lot less polluting, because sulfur and nitrogen free during the production of biofuels will not produce pollutants in the atmosphere. Now, so far we have been able to cope with the increase of the population, but this has caused a colonization of the Earth to a substantial amount. By 2050, we will be able, the culture farming will cover up to 30% of the Earth's surface. And the rise in efficiency is already not keeping up with the rise of the population. So we need to look for something different from that. And this is going to be, in order to face this, a number of challenges, not only for energy and food, but also to keep biodiversity high and decrease pollution and be able to have raw material for industry. Now, I don't know why it's not working. Yes. So we have in front of us two different attitudes. One attitude is to wrap our planet into a nice box and say, wow, the planet is wonderful, and we don't touch it. But this, of course, implies that the population stay like it is or goes down. Otherwise, we need to cope with that. And the way biotechnologists do that is to try to keep together the preservation of the environment and the preservation of the environment and, and increasing the productivity. This can only be done by devoting a substantial part of the surface to the biodiversity, and keeping the low biodiversity crops cultivation to a smaller area. This, of course, you have to engineer plants and algae to a higher productivity. And this is something that is very important, and this can be done based on the basic observation. If we look at the distribution of species and biomass into the Earth's surface, we see that, let's say, altogether, 50% of the primary productivity is on Earth surface, and 48% is on the sea. Of course, the sea is larger, but the concentration of biomass is lower because microalgae are continuously eaten by the other kind of organism in the sea, and therefore the balance between primary biomass but not productivity is moved to towards the sea. So the sea organism, the algae in particular, are a lot more productive than the plants that live on soils, but they are limited by the disponibility, the availability of light and nutrients. If we look into the distribution of nutrients and the distribution of light, what we see is that the light is available a lot on the surface, but nutrients are available underneath. And therefore, algae are strongly limited by the ability of the two sources for their growth. Now, if we grow them in photobioreactor, we immediately realized that are a lot more productive than our plants. And this is why we want to go for algae biotechnology in order to solve many of the problems that we have. The productivity is already high, but can be improved a lot. In fact, in a bioreactor, we have something that we already observed in the natural environment, which is light limitation and anoxia when algal bloom occur. So in a photobioreactor, the center of the photobioreactor is in the dark because the optical density is so high by chlorophyll that at the center you only get respiration, no photosynthesis, and at the external part you get too much light and you get photoinhibition of photosynthesis. But then when you can engineer algae in order to decrease their optical density, and you obtain a full photosynthesis active culture that is a lot more productive. We have done that in the past. We have obtained strains with small chloroplast which are twice as much productive of wild-type plants. And this is just an example. I don't want to elaborate too much about that, because I want to make a case, and the case is the one of chemical industry. The chemical industry is indispensable for social and economical development, and yet it is polluting because it is important for pesticides, polymers, for fertilizers. It has been essential for drugs, but it had a strong, we had to pay a strong toll for those facilities, which is pollution. Indeed, all the chemicals that we use in our society have been made from plants and from algae. Plants synthesize 200,000 chemical compounds, And all the molecules that we have been identified first in plants and algae, and then brought to the production by chemists with a very expensive procedure and very polluting procedure. Can we do something different from that? Okay, in fact, bioeconomy has been something that has been pushing the development of basic research for a long time. Von Humboldt and Bonpland, as much as Darwin, were paid in their travels around the world discovering new species and the evolution, for example, of the organism by the force of economy, because they were identifying plants useful for European productivity. And botanical gardens that we like very much, this is the one of Padua, this is probably the ancient in the world, still active, have been founded in order to keep those economically important plants around us, not for basic research. Now, this has been extremely useful for funding and for supporting the basic research by botanists and by biochemists, biophysicists, and this is still the case. This is the case that I want to make. One example is the case of isoprene. Isoprene has been discovered from the caoutchouc in the Hevea brasiliensis at the end of the 18th century. And from that, the tires and everything else, the plastic, let's say, have been produced by vulcanization and polymerization of this compound. Now, in fact, by copying that, Giulio Natta in 1938 got the Nobel Prize for the moplane, which is the polypropylene. He has been copying the isoprene for that. Because what can we do now? We can produce isoprene with isoprene synthase taken from poplar, and we can introduce in algae, and directly from the algae, expressing isoprene, from light and CO2 we can produce isoprene that get out of the cell and can be condensated in a cold trap and bottled straight away. No pollution, no high productivity, no waste in the production of this chemical compound. This is the model for the future chemical industry in the world, as many as for other purposes. But this is the possibility. Why we want to do that? And the case of ammonium synthesis is, would say, important in this respect. What we see is the Haber-Bosch on the left, the Haber-Bosch procedure that take atmospheric nitrogen and hydrogen from methane, and by a very powerful and power-consuming procedure at a very high temperature in 300 atmosphere produce ammonium. But the same ammonium can be produced at room temperature without high pressure by the diazotroph through the enzymes called nitrogenase. And the same amount, more or less, in the world at the moment is produced by chemical industry and that. With the difference that chemical industry explode, you know, every 5-6 years, one town altogether is blown up by the explosion of this kind of industry. And this is using a lot of fossil fuels. What do you do with agriculture when it will not be possible to produce ammonium? Well, we have to go for that. We go for the biological production of that. So essentially, beside that, there is one point that is important. Organic synthesis in chemistry produce byproducts, a lot of byproducts, and that are waste, in fact. While biological synthesis is a much higher yield and don't produce, because all the wastes are recycled into metabolism. So I would say that the synthetic biology, which is in front of us, is the way to go for having a sustainability in that. And I know how much time I have. Not much, so I will be as fast as possible, and I will jump over the technology that is well known by all of you that have been developed in bacteria for transferring genes and expressing genes, to the new things, which is the application of genome editing to unicellular algae. It is something extremely recent in the last 3 years, that now we are able to obtain high efficiency in transferring genes and knocking out genes and changing genes. And we can do some in a very precise way by new technology. I have to show you something that make the real difference. It's something that has been made in my laboratory in the last 2 years. We have been able to change 9 different genes in only one step of transformation in the unicellular algae. So this changed the game, because you can engineer a complete pathway, metabolic pathway, in one single step of transformation. So you can see that we use green gels, green proteins to knock, those genes were knocked out, and you see that from the wild type we got at least 2 strains knocking out all the genes encoding for the gene family. This is the most difficult thing to do when you have a multi-gene family and all the genes are very similar to each other. But now we can target each of them separately and with high efficiency. So the last point I want to make is that when we use bacteria, we use yeast for the biotechnology, you produce a lot of CO2, because the best efficiency ever that you can obtain is about 20%. So 80% of the substrate that come from biomass is going to CO2. But this is not the case of algae. Algae absorb CO2 and light and go directly into your product. So you will be absorbing CO2 into the chemicals directly and not producing additional CO2 with the fossil fuels. Now, I'm almost finished, because I want to tell you about what is new in the last days. It's probably not yet published, but I've been gathering all together. These things that has been put together in Chlamydomonas is now transferred to Phaeodactylum, Tricornutum, diatoms, and all type of algae. People is exporting the technology to a lot of algae, marine algae, and this is go back to the basic research, because it's possible that to make very precise variant of the populations, and use those variants in order to check hypotheses in the growth of the population in different environmental conditions. And this is already being done, for example, in Paris and in California and in Stanford, and give extremely good result with respect to the behavior of microalgae and autotroph in the environment in different conditions. So, and I finish here. I think that developing new technology is extremely important, and once you have made that in order to make economically important results, this will bunch back into your basic research and help you to understand the nature of our world. And with that I finish, and thanks the chair for allowing me a few minutes more.
Thank you very much. Thank you very much, Roberto. And now it's time for Frédéric Briand to deliver his talk. Frédéric is the Director General of the Mediterranean Commission, And so he has dedicated a big chunk of his life to foster the collaboration among the states in the Mediterranean, building a network of relationships that is precious to our scientific community.
Thank you, Nando. Good morning, everyone. I couldn't think really of a better way to start than this poster of an infamous movie. This is by far not the best movie, where Sophia Loren starred in 1960, but it says it all, at least to us marine biologists who work in the Mediterranean realm, because indeed it all started here, as we heard over and over for the past days. My own start in biology was with phytoplankton, and I'm going to sort of focus my presentation as Roberto, when he invited me kindly to be here, I thought I would develop some ideas about how we shape our views about what we call biodiversity now, which is really the way we see the world around us, and how it is organized, shaped. And this depends very much, the way we study it and our perception are very much interacting all the time. So I'll go through that through my own trajectory, if you allow me. I did my doctoral thesis in Southern California, and at that time, believe it or not, very little was known about the phytoplankton there in the coastal waters. So I had to draw on a Japanese book to identify most of the forms and shapes I was seeing through my phase microscope, and that book was remarkable, fortunately. And when you study week after week, month after month, phytoplankton, you unavoidably end up being sort of surprised by the enormous species richness that is in front of you on your microscopic plate. Just take this recent report from 2016, based on the Chesapeake Bay marine diversity. Phytoplankton, these columns exclude nanoplankton, microplankton, of course. But by far the richest component of that system is phytoplankton, with 1,453 species of phytoplankton identified there, compared to, say, 400 species of zooplankton in above 200 microns in the Chesapeake Bay as well, 348 species of fish, etc., etc. Again, this excludes bacterioplankton, nanoplankton. And so these same observations, of course, led a very famous man, which is a personal inspiration for my early years, which was named George Evelyn Hutchinson, who is certainly known to ecologists in this room, to write a famous paper towards the end of his life, which is called The Paradox of the Plankton. In other words, why do we find hundreds and hundreds of species of phytoplankton in the upper meters of the oceans and the lakes also, whereas the dominant theories would expect you to see competitive exclusion overrule, and in the end up with just a few dominating species. Why, after millions of years of evolution, do we still find hundreds of species in 1 liter of seawater? Okay, so that problem really interested George Evelyn Hutchinson through most of his year, and is pictured here in a lake in Tibet, sampling phytoplankton. When Matthew Frost gave his talk yesterday on the founding effect of the Stazione Zoologica Anton on many trajectories in marine biology, that inspired me to check in the background of Hutchinson, and believe it or not, I did find that as a young man, when he was 22, he left Cambridge on a Rockefeller Fund fellowship to spend 1 year here at the Stazione, where he worked not on plankton, but on the branch or gland of the octopus. And, but he left that later on, and he started to travel extensively and worked on lake and founded modern limnology, which is a study of lakes, and modern ecology, because there was a short supply of octopus at the time, in 1925. So this is how his story goes. Anyway, that problem drew my full attention for a number of years when I started my postgraduate career in Canada. And I installed a number of mesocosm, experimental mesocosm in lakes. And I manipulated different parameters like number of grazers, the instability of the water column, light intensity, carnivorous intensity. I added vitamins, I added nutrients, played around with all kinds of parameters like that to test the theory. I even went in a number of tropical lakes at different altitude to test water instability as one of the hypotheses, which Hutchinson also liked very much. And I pushed the thing to go as far as Himalaya myself, just to mimic my model. But that was on the other side of the Annapurna. Now, I got away from that and turned my attention to food web and other research. But in those days, there was a major revolution happening in the world of plankton. To illustrate it, I chose to do some cloud research on the words most associated with the term plankton in the thousands of publications that our Mediterranean Science Commission, CSM, has edited through the years. I mean, for the past 100 years, we've had a number of congresses where we have hundreds of participants, and we have digitalized most of those early papers, and so with keywords, etc. And so you see, when you type the word plankton, these are the words most associated with it that come out for the period, for the paper published between 1960 up to 1999. So it's very classical. You see zooplankton and phytoplankton coming up. And here, at the bottom, at the very bottom here, nanoplankton only take a very, very minor role. No mention of bacteria here. No mention, well, not that I see. I'd also, no, but well, that's true, because jellyfish, unfortunately, well, according to Nando, of course, jellies should come as big planktons, but they didn't come up in the top 50 words. And then, look at the association of the word plankton in the papers post-2000 that we published. And we have about 15,000 papers that were sampled. And here you find bacteria of carbon pelagic, of course, particulate matter. Phytoplankton and zooplankton are taking a minor role this time. And you also see Nemiopsis. Now, Nemiopsis, you probably know what it is. It's a comb jelly that was introduced from Florida by cargo ship in ballast waters into the Black Sea in the early 1990s, and where it totally destroyed the anchovy stock and the anchovy fisheries. 92%, 95% of the stocks were destroyed. Nemiosis ladyi is the name of this comb jelly. If you want to know more about it after I leave, you ask Nando, he knows much more than me. And it was a devastation. Eventually, that pest, so to speak, was controlled by another invader, which was called Bayreuth. Yeah. Yeah, I know. Anyway, so this reflects changing times, changing technologies. What had happened throughout those years is that our vision of how the biological world is structured changed considerably, and we welcome, along us, eukaryotes and bacteria, we welcome the huge group of archaea. And then this took another shape in the past 10 years, and now we are confronted with this incredible diverse vision of how the biological world is structured, with huge phyla of bacteria, archaea, and we found ourselves, around here, around these plants. This is, of course, thanks to huge advances in molecular genomics, et cetera, et cetera. Now you can fingerprint, an ID card, genetic ID card of 1 liter of water, and you get that. Wow. So, you get that. So, the paradox of the plankton has multiplied, so to speak. Because in one milliliter of seawater, you find coexisting around 10 million marine viruses, 1 million marine bacteria, 10,000 phytoplanktons, and 10 to 100 invertebrate or ectoplankton larvae. Wow. Wow. Again, this brings back to life the paradox of the plankton, the paradox of the nanoplankton. And in order to advance on this, you have to go back to your university courses in fluid dynamics and bring back the good old Reynolds number, which is a ratio, if you want, of inertial forces applying to the object or the organism that is moving through seawater, or through water, over the viscosity, the resistance of the liquid, if you want, where the numerator will depend on the size, the velocity, and the density of the organism. So if you are a whale, a marine mammal, density, size, high velocity, high Reynolds number. In the same liquid, on the other hand, a marine bacteria will have a very, very low Reynolds number. Here are the examples. So these are approximations, but they are consensual. So if you look at the column on the right, as you move up the size ladder from marine bacteria, Circum spermatozoid copepod, a human swimmer, a seagull, a large whale, you go from billions and billions of times of magnification. So when you are a marine bacteria, marine virus, or marine phytoplankton, to you, and it's completely counterintuitive to us. It's like swimming, in honey. It's really like that, You are or in jello. You are stuck in jello. So when you move, actually, you don't move. You bring with you your jello bubble, to the point that more and more of us now think that phytoplanktons, marine bacteria, viruses, are actually very rarely, if ever, in contact with each other. And yet, there will be no dynamics in this world if there was no vector for movement. And one of the, I will get back to that later, one of the vectors for movement is, of course, currents, and moreover, the ingestion of this plankton, which is often bycatch, I mean, incidentally. Like, think of a whale, baleen whale, you know? Baleen whale depends on the absorption continuously of tons and tons of seawater in order to get the krills and other things there. And along with the krill, you know, it will catch up billions of planktons. And if you look at this graph, which is a bit amusing, on the ordinate you have the sustainable swimming speed of the organism. So of course you have a fairly high speed for marine mammals like whales, fish also, about 4 kilometers per hour, and of course for zooplankton it's much less. So if you relate that speed of the predator to the length of your gut, the gut of the predator, it takes time. The transit time will be fairly long, if the gut is fairly long. And if the baleen whale, say, travels 50, 100 kilometers while you transit through its intestine, you know, you'll be released as a marine bacteria, as a marine phytoplankton, 100 kilometers later. That's one way, you know, you bring back some dynamics and interactions in the plankton. So these are my views. Just one back. I'm going to say something that you have to take with a bit of grain of salt. When I was working in the United States, most of modern ecology, evolutionary ecology, was being shaped by the students of Hutchinson, mostly. Odom, MacArthur, some Robert May also was a postdoc. These people have shaped our views of how the world, biological world, interacts, and most of it is based on an Anglo-Saxon, I would say, view of life, where it's a struggle and the survival of the strongest. You know, America is number one, etc., etc. And in those earlier, there were some very fundamental work by a great Russian scientist and also a naturalist and also an anarchist, Peter Kropotkin, who published in London in 1902 this very interesting visionary book called Mutual Aid. So this is about what we call today symbiosis, mutualism. Since he was coming from a very chaotic times also, the revolutionary, pre-revolutionary world of Russia, his views were framed and shaped by other driving concepts. And the more you think of it, the more it seems, and Hutchinson also was aware of that we have to probably integrate more and more of those forces into the way we see the world, the biological world. This is an example, you know. Macroplankton depends on bigger organisms to be carried away, okay? We know that with pollinators and plants, but this is much more profound than you think. And so that's the first point I wanted to make. A second point, which will be briefer. Now I turn to whales. Whales, as I said, larger organisms, very high Reynolds numbers. To them, swimming, you know, is no effort. think also of other cetaceans like dolphins. It's no effort, because there's hardly any resistance from the water. So, thanks to that, they can undertake huge annual migration between their feeding ground in polar waters to their breeding grounds in tropical waters, where they will mate and give birth. If we focus just on just 3-4 species of high migrating whales, you have, for example, the gray whales and north right whales in northern hemispheres, been moving from Arctic to tropical waters, and in the southern hemispheres, the southern right whales. And then in both hemispheres, you have the humpback whale, very, very well known to most of us. Humpback whales, for example, they, you see, yes, okay. Well, they go, to Madagascar, the Réunion, and back and forth. Except that in the past few years, we have seen very drastically reduced numbers of humpback whales going back to their breeding ground around the Réunion Island. And so this poses a question of the vulnerability of the largest animals now, of course, around us, living animal. And of course, the cause is us. There are many reasons for that. I mean, it could have been, whaling, but now hunting has fortunately much recessed. And now the major causes of whale and cetacean death, I'm talking of thousands and thousands of individuals every year, I mean hundreds of thousands, are cut in half by ferry ship or cargo, are stranded because of the huge noise underwater caused by maritime traffic and also drilling operation. And it's like us sitting in a discotheque by the, ampli, just with a heavy metal band around. I mean, you just cannot talk to each other, And these animals depend on that for their survival. Entanglement is in net. Drift nets, abandoned fishing gears. These mammals are mammals. They have lungs. They have, even though they are very good divers, sometimes they can stay underwater for up to 2-3 hours, they have to go back to the surface from time to time to take a breeze. And then they are entangled, they just drown, unknown to us, because it's just underneath, the water surface. And so there are huge operations now by some NGOs to untangle them, but still, the statistics are enormous. And so you add to that epizooty, viruses, etc., which are going to increase because of the increasing water temperature. Climate change, which is going to change the shape and trajectories of stream jets, like the Gulf Streams. Where are we going? We are going to the accidental death of the oldest organism in the pod that knew the routes of migration. That's a very interesting thing. How do you think they know the route? I mean, how do you think they can travel from Alaska along British Columbia all the way to Baja California, for example, for the gray whale? And how do they learn that? There must be some cultural transmission, okay? And so if by accident some sort of drift, if you want, genetic drift, you lose the one organisms, the elder who knows that, you may lose the root, okay? So there is some hope. I mean, that we have underestimated the capacity of some of these species to communicate over hundreds of miles among themselves. So we have a meta-population of humpback whales in the South Pacific, you know, on the eastern shore of Australia, all the way to French Polynesia archipelago and Samoa. And what recent research has discovered is that they transmit some new songs amongst these different subpopulations, okay, from west to east. So there is still some culture, some acculturation and some learning. So, one second lesson, connectivity, okay. We don't invest enough in the study of whales. We leave that to some powerful NGOs, okay? We lack funding, interest, although there is enormous population interest for these organisms. But this is a race against the clock, okay? I will finish with one last animal, on which I'm working now with my group, some of my collaborators. This is a Yelkouan shearwater. This is a shy, timid, poorly known seabird. It's an exclusive maritime bird. It nests. It never flies over land, over shores, never. It nests on uninhabited islets away from us. And what we know of some nesting sites of Malta, Croatia, Tunisia, Greece, Turkey, in the Mediterranean Sea. It has also been observed sporadically in the Black Sea, around Bulgaria, the way to Crimea. No nesting site there. We don't know, we just know that it leaves and comes back every year. It leaves its nest in burrows and it reappears, okay? So now with new technology, I mean, very small light GPS satellite transmitters, we have decided to focus on this seabird, another example of an organism which is not sufficiently studied in marine stations. They're just a fashion victim. It was never much studied. We left that to you know, ornithologists, weekend ornithologists, you know, they make records and they compile their notebooks and there is very little thing on this bird. And last year, we equipped, this is one of their nesting sites in the natural reserve Lastovo archipelago. It's not too far from Split, but far enough, where they have burrows. And so we equipped a dozen Yelkouan. And this is a young chick, just come out, okay? And adults, and also 2 sweet chicks, just to see. The transmitters are about 8 grams. And we followed that. Well, one bird took off a few weeks after we equipped them on 7 July. 2 weeks later, we were following it, you know, hour per hour. We had satellite transmission. 2 weeks later, it went around the Peloponnese, and there we didn't know what he would go. Would he go to Libya, or would he go north? It stopped around some other islets south of Athens for about 2 weeks, and then it moved north decisively, northeast, and on 10 August, it was there. And then it rested just briefly. You could see when it rested because it just sort of sleeps over water like a duck, you know, and so you just see strass on the radar screen. And in 24 hours, it crossed the Dardanelles. It slept over the Marmara Sea about 6 hours, and then at dawn, in 40 minutes, very quickly, it went through the Bosphorus because on both sides there was Istanbul. And I can see how he must have been panicked with all the traffic, tankers, lights. And then we lost its trajectory. About 10 miles away from shore. We lost its trajectory because all the satellites were bugged and things, and we retrieved it 3 weeks later near, to the east of Sevastopol, to the south, I mean, near the Strait of Kerch, near Novosibirsk. And when we retrieved its signal, we retrieved its path. We had access to the data, the transmitter. So we saw that it spent 3 weeks happily in the middle of the Black Sea. And then it stayed there until we lost the signal, because in the end, this bird dives, it can dive 10 meters underwater, and so you see the attachment of the transmitter eventually is lost. This is activated by solar battery, but eventually we lost the signal. Luckily enough, this bird was joined by 2 other of our 10 birds. So in total, 3 adults undertook the same journey, but independently at different times. And 1 young chick, 2 months old, also undertook the journey. Okay, so last December, I sent one of my collaborators to Novosibirsk. This is the foot of the Caucasus there, to work with Russian professor of the University of Moscow. And those are just yelkwans there that you see. What are they doing? Okay. This is my final slide. What are they doing there? Well, they are just, why undertaking a journey of 3,200 kilometers, you know, because they feed on young anchovies, which are, you know, spawning grounds of anchovies. The Sea of Azov is a big spawning grounds for anchovies. Later on, they migrate south, okay? They will go to the northern shore of Turkey, and this is where they replenish, you know, all their proteins and things, and then before undertaking the journey back to their nesting site. So I find that as a powerful, of course, metaphor for interconnections between system, cultures, human beings. We have left sort of protocol and studies methodology to local fishermen there to alert us on the next arrivals, etc., behaviors. We work still closely with Russian scientists, of course. And so this is another call. This is my last point. I mean, regardless of the chaotic times we traverse, I mean, we have to remember that we scientists have very little to do with the way nations are governed, and the last thing to do is to boycott our cooperation with people on the other side, as we said. And we have the same enthusiasm on the other side. Okay, so Russia is one of the good members in our commission, and it's also a call to study organisms that are poorly studied, to go where few people go. You have to keep that enthusiasm and that curiosity. This is what makes us scientists, you know. Thank you.
Thank you, Frederick. And now I give the floor to myself. The chair of this session was supposed to be Roberto Da Novaro. Roberto Da Novaro is not here, you might have noticed, because he is in Palermo, where Italy is trying to build a national center on biodiversity. And so is there representing the zoological station and so on. For the first time, Italy is recognizing the value of biodiversity. And we are investing a lot of money on this topic. And my talk will be also related to this, and also to something that Frederick was mentioning at the very beginning. Evelyn Hutchinson, besides The Paradox of Plankton, published also the Homage to Santa Rosalia. And it did it while he was visiting Palermo, so it was in Italy anyway, and he found in a pond more species than he expected. And so he asked why there are so many species, which was also the paradox of Plankton. Another eminent ecologist, who was a physicist, Robert May, who became president of the Royal Society in London, published a seminal paper in 1970 entitled Simple Mathematical Models with Very Complex Dynamics. And this is the usual attitude of the people in physics who arrive to biology and with some equations pretend to explain everything. After 10 years, he published another paper entitled How Many Species Are There on Earth? Because you can have all your equations, but if you don't have the variables, the equations are meaningless. And so he asked for the exploration of biodiversity. One thing is to make the inventory, how many species. Another thing is to ask the question why there are so many species, and so on. And these are still problems that are prominent in our scientific community. And they were thought for a while to be old, but now we are investing a lot of money in order to answer these questions because they are crucial for our survival. And so we are passing from agricultural oceanography, which explains everything with nutrients, to behavioral oceanography and ecological oceanography and so on. So Darwin and Dohrn, first, and this is the Darwin-Dohrn Museum, were zoologists. And they started their careers by writing monographs. The one of Darwin was on barnacles. The one of Dohrn was on pycnogonids or pantopods, sea spiders. And so before building big ideas, they were answering the question, how many species are there around in some groups, because they had to know biodiversity very well. And so they were mastering this field of biology. Darwin founded evolution and ecology, and I dare to say that Dohrn founded modern marine biology. The two souls of the Stazione Zoologica, we have heard it yesterday too, were zoology and physiology. And so there was, zoology was not only zoology, it was also ecology and botany and so on. So let's say today we would call it biodiversity. Or better, marine biodiversity and ecosystem functioning. The other one was physiology. Again, it's not physiology anymore, it's molecular biology, it's biotechnology, it's lots of new things that are extremely important. The two visions were equally important. And so in the vision of Anton Dohrn, both these research programs were worthwhile being accomplished. But the most ambitious goal of Anton Dohrn and the zoological station was to answer the question, how many species are there, what they are, what they do. And so he launched the monograph series of the fauna and flora of the Gulf of Naples. And you can see it here. This is a group of 39 volumes plus one, we will see why, that started with something that goes deep in the tradition of Naples culture, because the project was built while standing on the shoulders of giants, and the giants were people that, no, the other way, come on. I pressed the button, but Naples had a very long tradition of marine biology and exploration of biodiversity long before Anton Dohrn. It was a prominent European capital, but it was also the capital of marine biology. And the systematic exploration of biodiversity started in 1758 with the Sistema Naturae by Linné, flourished at Naples with Neapolitan scientists like the ones you can see here, but there are many others, Spagnolini, Delle Chiaie, Cavolini, of course, and so on. All these guys were writing monographs on the fauna and flora of the Gulf of Naples. That one, Stefano Delle Chiaie, was hydrophytologie, so he was studying the microalgae and the algae of the Gulf of Naples. An important person is also this one, Salvatore Trinchese, because Salvatore Trinchese was a specialist of nudibranchs. He worked in Genoa, in the same institute where I took my degree, much earlier than myself, and he published this beautiful monograph on the Eulidiacea, which is nudibranchs of the harbor of Genoa. From Genoa, he moved to Naples, and he was rector of the university for 2 mandates. And he's the one who gave the honoris causa degree to Salvatore Lobianco. So there was a big link between the university, the Federico II University, and the zoological station. So we might say that the seed of the Stazione Zoologica was set in very fertile soil, because there was a pre-adaptation of the cultural territory to embrace the project of Anton Dohrn. This was the first monograph, 1880. It was on the comb jellies, like the Nemiopsis that Frederick was talking about. The station was founded in 1872. In 1880, the first monograph was launched. Writing a monograph like that requires years and years of work in the field and then in the laboratory. And so Kuhn started this series in 1880. The last monograph is this one, 1970. The hydromedusae of the Gulf of Naples, of the Mediterranean. Anita Brinkman-Voss. I spoke about that yesterday when I said that Rupert Riedel was supposed to write this monograph, but then it was Anita who made it. And she made only the first part. So there is Antomedusae attecate, and then there are part 1, Capitata. And then there are much more. I finished this work in 2008 with a monograph on the hydrozoans of the Mediterranean, and to write a book like that, you need to work 20 years. So it's not an easy task. After this one, there was another one on the opistobranchs, and the figures here are of a genus that has been dedicated by Salvatore Trinchesia, okay? Made by Schmeckel and Portman. This was not, even if it's written on the book, was not a real part of the series. It was printed in a different format by a different printer and so on. But anyway, in '82, there was the monograph. Now, let's go into the, come on. No, okay. There is a lag between pressing the button and, okay. So the zoological station with these monographs was and not only the zoological station, Naples with the zoological station was the world capital of marine biodiversity. With the monographs and also with all the species descriptions and so on that had been published in the journal. In 1992, with the Rio Convention, where Frederick met his wife, it's an historical date also for you, it was universally recognized that biodiversity is vital for our survival. And that the erosion of marine biodiversity is our problem number one. And there is not a problem number two. If we keep going on the way we are doing, we are cutting the tree, the branch of the tree where we are sitting. So in 1992, the central role of biodiversity was universally recognized. In 2011, 2020, it was the decade of biodiversity for the United Nations. The European Union, with the Marine Directive, the Marine Strategy Directive, identified biodiversity as the descriptor number one of good environmental status. Naples was the world capital of biodiversity. They stopped producing monographs on biodiversity. I have only one word to label this behavior: stupidity. But describing species was not enough. Salvatore Lobianco, whose laboratory has been reconstructed there, published this very important monograph on the biological notices on the sexual reproduction of the animals of the Gulf of Naples. That was the catalog of experimental animals for those who were coming here to conduct research on general biology. So they were using these animals to uncover specific problems in the field of physiology, okay? And he made this inventory, species by species, based on the monographs that he contributed to write, saying where these animals can be found, when they are reproductive, what are the habitats where they live. So he was studying the phenology of the biodiversity that was going to be described. And so there was the biodiversity and a beginning of the study of ecosystem functioning. This model has been adopted at other stations. For instance, this is Plymouth. And the Plymouth marine fauna did exactly the same thing, species by species, where they can be found, when they are fertile, and so on. And this is La Faune Marine de Roscoff. Again, exactly the same things. And this is Woodsole. So in marine stations, and this is the network, they were inspired by the work in Naples to do the same and build these inventories. Now, the descriptor number one of good environmental status of the European Union is biodiversity is to be maintained. what is the benchmark? What is the state of biodiversity that we want to maintain? How can we measure the efficacy of our interventions in order to have sustainability? We need to maintain biodiversity, but we need to know how it was before. And there are only 4 places on the planet where this has been done, starting with Lobianco 1909 to the inventories in Roscoff and so on. So the project should be to check the state of biodiversity now against the known state before. I gave a seminar to Roscoff and I asked them, but do you know what is present outside here now? Because you have your inventories, but they are 50 years old. How did it change? They don't do it, okay? They don't do it. They don't do it anymore. So you have a facility like this, it can answer problems that are extremely important, they are recognized as crucial for humankind, and they do other things. And nobody is doing what they would be supposed to do. So marine stations in the history of the zoological station are supposed to do these things. The other, besides the monographs, the other publication of the zoological station was the Mitteilungen that then became the Pubblicazioni della Stazione Zoologica. And so the title shifted from German to Italian in 1916. The first war, and we were not in good terms with countries where German was spoken. In this journal, both the zoologists and the physiologists were publishing the outcomes of their studies here. So it was the journal for both communities who were working together. Another thing that Dohrn wanted was the communication among the different scientists in order to exchange ideas and so on. And so, for instance, Hutchinson came here to study the nervous system of octopus, and then he became an ecologist. So there was a permeability among these different approaches. But this attitude stopped more or less when the impact factor became a way to measure the quality of the work of researchers. And zoology had a tiny impact factor, and physiology and all the rest had huge impact factors. And so there was some sort of conflict between the two approaches. Here there is an historical thing that should be said. The Institute for Scientific Information that was giving the impact factor was giving also another measure of journals, the cited half-life. For how long a paper published in that journal keeps being cited. And if it's more than 10, means infinite. If you rank the journals, the higher the impact factor, the lower the cited half-life. This gold in the high impact factor journals becomes, I don't say the word, after a while. Whereas the silver of other remains silver forever. And if you describe a species, that description remains forever in the scientific community. The monographs of the zoological station keep being cited, and they are still very important because they are the only ones. The physiological papers published in those years are completely forgotten, or they are used only for historical reasons. So the journal was closed. And as I said yesterday, talking about Rupert Riedel, it was Rupert Riedel who rescued the journal and transformed it into PSZN1 Marine Ecology. PSZN means Pubblicazioni della Stazione Zoologica, and it was written in the cover of the journal. If you look at the authors, none of them used to work in Naples. Because zoology and so on, biodiversity was going to be dismissed together with the journal and the monographs. Stupidity again, because this was considered as the problem number one for our species, and the capital of the study of this problem number one was dismissing this field of research. The journal changed name several times. You see the second step where it's written again, Pubblicazione della Stazione Zoologica di Napoli. Then it became an evolutionary perspective, marine ecology and evolutionary perspective, and then now it's simply marine ecology. So this gives, I don't think it's right. I think that also what was called physiology should have the same dignity of this one, okay? But it was not considered as an important thing to have a journal dedicated to that. And so marine ecology remained. But today in the zoological station, both approaches are tackled, recognizing the project, the validity of the project of Anton Dohrn that wanted the two approaches to be together, working in side by side without any complex of superiority by one against the other. And so this is the new journal. The editor-in-chief is Roberto da Novaro, who is playing with lots of balls in the air, trying to keep the machine going, and is doing it very well. And now is in, as I said, in Palermo for the National Center on Biodiversity. There was also another marine ecology was called PSZN1 marine ecology, but there was also PSZN2 history and philosophy of the life sciences. And so this was the other journal of the zoological station, which is still a journal of the zoological station, but we don't have the subscription to it, so we cannot see it. And the history, other publications are miscellaneous publications like this one, the history of the Stazione Zoologica Anton Dohrn, that you can take for free in the bookshop of the museum, because there were tons of it. They wanted to throw it away, and I said, no, it's an historical thing. Let's give it to the people who come visiting the museum. And here are the other miscellaneous publications of the station, like the activity reports that are extremely important to reconstruct the activities of the zoological station. There is the beautiful book by Christiane Gröben on the correspondence between Charles Darwin and Anton Dohrn that inspired the building of this museum. There is a little monograph on Posidonia, because for a while the Ischia Stazione, the Ischia laboratory was the world capital of the study of Posidonia, biology and ecology, with the work of Lucia Mazzella and many others who came from all over the world to study the Posidonia meadows in Ischia. And now, luckily, with the work of Gabriele Procaccini and others, this prominence is coming back to the Stazione Zoologica where it belongs. Another publication of the Stazione Zoologica, you can see the symbol of the Stazione Zoologica on the logo on the cover, is this Mickey Mouse book. And you can see Goofy, for instance, up here, who is speaking about marine snow. Yuk, there is also snow. And in this museum, there is the marine snow installation that you might see if you want. And there are many things in this comic book that are present in this museum. And there is also Dornius, who is rescuing Mickey Mouse with his submarine. So another publication will be published within this year, we hope, with beautiful illustration. It's the history of the immortal jellyfish. The immortal jellyfish is Turritopsis dohrni, and the story is that this immortal jellyfish, described by Weismann. Weismann is the one who founded neo-Darwinism with the separation of the germ line from the somatic line. He was also a zoologist, and he described this new species that he called Dendroclava dohrni, and then the genus was changed into Turritopsis dohrni, and this species can reverse its ontogeny, and it has been labeled as the immortal jellyfish. The authors were very much impressed by this story, and so they built the story on one of these jellyfish that is taken, because the immortal jellyfish has been described from material coming from the Gulf of Naples, and so they tell the story of the zoological station and of Anton Dohrn, told by a jellyfish who studies the humans and reconstructs the life of, and you can see Anton Dohrn walking on the sea bottom with his umbrella, making his projects of the zoological station. So today, marine biodiversity and ecosystem functioning are the pillars of the European directives on the ocean, including another mission that is healthy oceans, seas, coastal and inland waters, where again, marine biodiversity and ecosystem functioning are considered to be crucial for the preservation of the integrity of the habitats that support us. The knowledge stored in the publications of the Stazione Zoologica is the benchmark to measure the efficacy of our initiatives. So something is sustainable if there is no erosion of biodiversity, and we have to match what we are doing with the natural conditions, and we have to know them. And so we have a treasure of information here that there's no match anywhere else in the world. And it would be crazy to do something different than this. I must remember that here, one of the great accomplishments was the Genome of Tomatoes, okay? The Genome of Tomatoes in the zoological station. You do it in an agronomic institution. Here we have to do other things. Or anyway, we have to do also the biodiversity and so on that was dismissed completely. So the mission of knowing these things is not accomplished yet. The ideas of Anton Dohrn are still extremely actual, and the premise for a knowledge-based approach to marine sustainability are in the history of the zoological station, and also the future of sustainability is in the zoological station. I might say that the dream of Anton Dohrn is being realized now from the biological station, because the zoological station opened new seats in Sicily, in Calabria, in the Marche region, in Latium, in Ligury, and so on. So the network is budding from the zoological station with a vision that is extremely modern, putting together biotechnology and marine biodiversity and ecosystem functioning and conservation. And so exactly on the lines of the project of Anton Dohrn. Another thing that has been done is the recruitment of dozens of young researchers who are carrying out this research project. And so the future is signed with the center, the National Center on Biodiversity. This will be fostered further, and the zoological station keeps being the capital of marine biodiversity and ecosystem functioning as Anton Dohrn designed it. Thank you very much. And now there should be the conclusions by Christiane, who should be with us. So Christiane, I give you the floor and to make the conclusions of this meeting.
Thank you so much, Nando, and of course I'm already looking for my papers. Thank you, all the speakers of this morning, and it's a great pleasure. Well, yes, it is a pleasure to conclude such a wonderful meeting with so many stimulating talks, and of course it's also a sad moment because it's finishing. But what I would like to share with you is also something I mentioned several times during my talks, and what I would have liked also to let you hear in original, but it's not possible. So this is one of the aquarium songs that have been written by Angelo Andres. He was an Italian zoologist from Milan, working as head of the library department at the Stazione also, and he wrote one of the songs in 1885 that specially has been sung also on the excursions on the steamer of the Naples station. And it went, but I won't let you hear my voice on the melody of Tiritomba, a very famous Neapolitan song, very much loved by the German tourists in particular. I never found out why, but anyway. So let me just read the text. We tried to translate it into English. Towards the beaches of the Tyrrhenian Sea, Anton Dohrn one day departed, and in the Bay of Naples he built the station. From the height of his terrace he spoke and wrote to all the world, "Oh, scientists, hurry to come and see my palace." Said and done. To the station came running, as they were a single person, professors from all nations, from all languages and regions. Russians, Yankees, Dutch, Belgians, Swiss and Prussians, British, Spaniards and Hungarians, Danes, Bavarians and Italians. Each of them strives to discover the great secret of life. They want to see the embryo appear, develop, and then die. And they use sublimate, osmic acid alcohol, glycerin, biochromato, or in case, gold chloride. Each of them wants to study the great mystery of the life of shark and torpedoes, but take a crab with a great treasure. Here they come, all driven by the mission of science and of work. They all rush to come, all those professors. And then they print in learned papers that they have not discovered the great secret, but that with better tools and methods, in future they surely will find them. and later they bring to the end of the earth, dispersed here and there among the people, a new civilization. And with this, the fame spreads out and runs as fast as sound, and all over the world, everybody invokes the Zoologische Station. So thank you for this. I'm so sorry that I can't hear you or reactions or whatever, so I just go on talking. And I also want to tell you that there has been a message sent to me and to Roberto da Novaro by Peter Callan from Australia. He's the brother of Amaryllis, and he wanted to share, at least in a message with us, this special meeting. And so it comes to an end, also with thanks to everybody, to the speakers in first place. And I'm very great and happy that the general lines that we started to plan about a year ago in a special group called 150s, where we planned this meeting and where we wanted to start with the beginnings with Anton Dohrn and going through times and finishing up in the future and in the projects we have here and dreams we have at the Stazione. And what came out to me or remains with me right now is the impression that the networking that Anton Dohrn planned, and this is why he called his station a station, like a railway station, still works and still functions and still is considered necessary to join scopes and tasks and to do something for our world to let it survive. So I very much encourage you all to follow also the other events. This group has been planning for this not birthday but birth year of the Stazione Zoologica Anton Dohrn. And all my thanks go, I can't name everybody, and some of the people who have collaborated, I couldn't even meet them, also because of COVID and under other commitments on my side. I've just participated in the 17th Naples summer school on the island of Ischia, but we had to interrupt it for COVID. But this is also a long tradition that Bernardino Fantini also mentioned, and several other people. So thanks to everybody who did, from the technicians to the, whoever made it comfortable for you to stay in the Dohrn Museum, the splendid Dohrn Museum that we have now. Thank you for all those who gave their thoughts and their manual work and their contribution. And not because we have the same name, but thanks goes on one name, but to all those who have given her a hand, to Margherita Grippen. And the group which is Naples, everybody. So please forward the thanks to everybody. And it was such a pleasure having you. And all my best wishes go also to Tim Hunt for recovery very soon and fast. And thank you so much for this great event. And I'm very grateful that at least I'm well enough, or I'm really perfectly well, to be able to take part in this meeting by Zoom. I'm looking at you right now, and I wished I was on one of the free seats among you. That couldn't be, but thank you for having been there. This has been a pleasure and a satisfaction also of the work I dedicated for many, many years to the history of the Naples station. And it makes me feel it's worth going on and passing also what I learned on to others who are collaborating right now. Thank you so much. Have a nice and safe trip back home, and see you soon on another occasion. The next birthday, we'll find an occasion to see you again. So bye-bye from the Naples station, and thank you so much.
Ciao Cristiane, recover quickly. We look forward to having you with us. And so the celebrations are over, and it's okay, you can go. But you can go, because if you are interested in having a visit to the museum, those who want, can remain here, and we can have a visit. Yes, and I will guide a visit to the museum for those who are interested. The others can go. And ciao. So, if you, let's see, and nobody remains.